Hello students, today we are going to start with our lecture on pollen pistil interaction. So हम लोगों ने इससे पहले पढ़ा है कि pollen pistil interaction, this is the last event or the terminal event of pre-fertilization. तो आज हम इसके ऊपर डिस्कस करेंगे. So as you see that pollen pistil interaction is already given as title. तो हम लोग आज जिस पे बात करेंगे कि पोलन आपको पता है कि दिस इज द मेल गैमेटोफाइट ऑफ एंजियोस्पर्म एंड पिस्टल दिस इज अ स्पोरोफाइटिक पार्ट of the female reproductive structure which is carpel. So in a way what we see is that it is a gametophyte to sporophyte interaction, right guys? So what we see that this is a gametophyte to sporophyte interaction. So we see that this gametophytic part, this is your pollen grain. So we see that pollen grain, this is the male gametophytic part of angiosperm, and when we say that we are talking about a sporophytic interaction, so this sporophytic part here is represented by the stigma, right? So talking about the pollen pistil interaction, what we see is that pollen pistil interaction is a process of continuous chemical dialogue. So this is a statement what we see in NCERT, that it is a continuous chemical dialogue. So बच्चों हम लोग क्या देखते हैं कि जब डायलॉग्स होते हैं, इट्स अ कन्वर्सेशन, राइट? इट्स अ कन्वर्सेशन बिटवीन two parties, two ends. So what we see here, why we are calling it a chemical dialogue, through chemical signals we will see that male gametophyte will now have a conversation with the female gametophyte, and we all have studied before and I am going to mention it again that the female gametophyte here is our embryo sac. So it is a continuous chemical dialogue, which means that there will be chemical crosstalk between pollen grain and embryo sac, right? So what we see is that in this time when we are talking about pollen pistil interaction, this interaction will start by the event called crowding effect. So we are going to now look at what is crowding effect. So in a very simple language, if we try to explain what is crowding effect, it is basically saying the more the pollen grain which will accumulate on top of the stigma, the better will be the germination. So it's a very simple idea which is saying that if we are looking at a stigma head, let's imagine this is the stigma. So we see the more number of pollen grains which will get accumulated, better will be the germination. So what we see that if there are more number of pollen grains which are accumulated on top of the stigma, those pollen grain will help to absorb the water vapor, and then they will secrete their enzymes better, and you will see that adequate amount of enzymes and salts and sucrose will be present, and that will help in the germination of all the pollen grains. So we see that accumulated pollens will absorb water vapor, secrete enzymes, sucrose, salts of boron, calcium, magnesium, etc. And this will lead to the germination of pollen. So this is the first start or the initial event what we see in pollen pistil interaction. So we will see that this incident will lead to the germination of pollen. So now we are going to talk about the pollen germination event. So what we have seen so far is that in our previous classes we have discussed that a pollen grain has one germ pore, or it may have more than one, two or even three germ pores. So these germ pores are generally blocked by the presence of callose sugar or callose, what we call as callose plug. So if we draw the diagram, this is the inner sporoderm, which is called intine, and we have a thick and discontinuous external sporoderm, which is called exine. So the portion which is discontinuous in exine, that portion what we see is blocked by the presence of callose sugar. So we see that this callose sugar has to be degraded for the pollen germination to occur. So we see that by the help of callase enzyme, callose plug will be removed. Once the callose plug will be removed, we see that this portion, the germ pore portion, will become available for the pollen germination to take place. So initially what we see that the inner layer or the intine layer, it will extend so let's imagine this is the inner layer or the intine layer. So this will extend, whereas we see that the external layer or the exine layer, which will remain as it is. So this is the exine layer which will remain as it is, and we will see that the internal layer will extend, and that will give rise to the structure of pollen tube. Okay. So what we see that if we talk about pollen tube, as this is an extension of our intine layer or the inner layer, we see that pollen tube, this is pectocellulosic in nature. and we see that this pollen tube will now carry three things. So this pollen tube will now carry our two generative cells and one vegetative nucleus, right? So we have seen this in the development of male gametophyte or the development of pollen grain, that the pollen tube will carry two generative cells and the pollen grains, they carry also one vegetative nuclei. So all those materials, the pollen grains, what they had before, the generative cells and the vegetative nuclei, they will now come inside the pollen tube. So what we will see next is that inside the pollen tube, the terminal position of the pollen tube, if we talk about the terminal part or at the apical location, so what we see, there will be the presence of a nuclei which is having a irregular shape. So this nucleus is your vegetative nucleus, which was previously known as vegetative nucleus. Now onwards we are going to call it as tube nucleus. So this is your tube nucleus. So tube nucleus, what it is? It is basically the vegetative nucleus. So this tube nucleus, once it comes at the terminal position on the tube, it will start forming callose septa. So once they form the callose septa, what we see that it will restrict, the septa will restrict the flow, the backflow of the tube nucleus towards the other part of the tube. So ये जो आपका septa बना है, ये septa जो vegetative nucleus है या tube nucleus है, इसको पीछे जाने से रोकेगा. At the same time, we also see that they will carry their generative cells in the subterminal locations, and once they keep the generative cells in the subterminal location also, we see that they form the callose septa, and these callose septa also will restrict the backflow of this generative cell. जैसे हम लोगों ने देखा है कि आपके पास जो ट्यूब न्यूक्लियस था, वो ट्यूब न्यूक्लियस पहले हम लोग इसको वेजिटेटिव न्यूक्लियस बोलते थे, एंड हियर ऑल्सो सिमिलरली जो आपका जेनरेटिव सेल अभी बोल रहे हैं, मतलब पहले बोलते थे, नाउ वी आर गोइंग टू कॉल इट एज स्पर्मेटोजोइड्स. So spermatozoids, initially they were known as, we used to call it as, बच्चों बताओ, so we used to call it as generative cell, right? So you know that these spermatozoids, these are basically your non-motile male gametes. There are two more things we will talk here regarding the cytoplasmic area of tube nucleus and also the cytoplasmic area of our spermatozoids, or what you call as generative cells. So we see that here the cytoplasm of the tube nucleus, we call it as active cytoplasm. So this active cytoplasm, this is known as active because of the reason that this cytoplasm carries auxin it carries cytokinin, and it carries hydrolase enzymes. Okay. On the other hand, no such thing will be present in case of the cytoplasm of these male gametes, or you call it as spermatozoids. So we see that these cytoplasm, they are known as inactive cytoplasm. सो ये जो एक्टिव साइटोप्लाज्म आप लोग जो देख रहे हो कि ट्यूब न्यूक्लियस के साइटोप्लाज्म में बन रहा है, ये एक्टिव साइटोप्लाज्म का फंक्शन क्या है, क्या फंक्शन प्ले करता है, दैट इज समथिंग व्हाट वी आर गोइंग टू डिस्कस नाउ. सो वी सी दैट द हाइड्रोलेज एंजाइम which has been formed by the tube nucleus in the cytoplasm, we see that this hydrolase enzyme, it helps to break down the transmission tissue of style. So this we have studied before, that the female reproductive structure carries a stalk like portion which is called style. और style जो है आपका, these are of two types. We have solid style, right? We call it as solid style. And we have hollow style. So the style portion which is solid, you see there is the presence of a tissue in between. So this tissue, this is known as transmission tissue. Right? So what we see that the pollen tube has to grow through the style. स्टाइल के अंदर से पोलन ट्यूब को ग्रो करना है, एंड टू ग्रो थ्रू द पोलन ट्यूब the cytoplasm of the tube nucleus, which is now carrying the hydrolase enzyme, it will help to degrade the transmission tissue if the plant is having a solid style. अगर प्लांट के पास hollow style है, in that case the transmission tissue is absent. So in that case hydrolase enzyme का style के ऊपर कोई function नहीं है. What we will see that these hydrolase enzymes will again act when it will going to finally interact with the embryo sac. So we'll go there. So we see that auxin and cytokinin, these two plant hormones are also being produced in the active cytoplasm region. So this auxin and cytokinin, this works as a chemical signal to activate the ovary wall. Okay, and it also acts as a chemical signal to the embryo sac. So we will study this in our future lectures, that this incident, when we see that auxin and cytokinin, they are working as a chemical signal to activate the ovary wall, this particular incident is very important for the formation of fruit. So what we have seen here is the first signal for fruit formation. We'll come back and revisit this section when we will talk about fruit formation. But here our main target to understand that how auxin and cytokinin, they works as a chemical signal for the embryo sac activation, or you can say as a signal transformation to the embryo sac. So what we see here, the very moment the embryo sac will receive the chemical signal from auxin and cytokinin, embryo sac becomes activated. Once the embryo sac becomes activated, we see that this is the situation of the egg apparatus which is present in the micropylar end of embryo sac. So we are now just looking at the egg apparatus. So you know that egg apparatus जो हमारे पास होता है, यहाँ पे 3 cells आपको दिख जाएगा. There are 2 cells which are having the finger-like wavy cell wall. This is known as synergids, right? ये हम लोग पहले lectures में पढ़े हैं कि the cell which is having the finger-like projections in their cell wall in the micropylar end, they are called synergids, and the cell which is present in between, that is your egg cell. So once the embryo sac becomes activated, what we see, one of the synergids will now degenerate. ये जो साइनर्जिड है आपका, दोनों में से कोई भी एक साइनर्जिड जो है वो आपका यहां पे डीजेनरेट हो जाएगा. सो लेट्स से फॉर एग्जांपल्स मैंने जहां से मार्क किया है, दिस साइनर्जिड इज नाउ गोइंग टू डीजेनरेट. सो डीजेनरेट जब होता है, देन यू सी दैट आफ्टर डीजेनरेशन इसका जो फिलीफॉर्म एपरेटस है, फिलीफॉर्म एपरेटस कौन सा वाला है, जो आपका वेवी सेल वॉल है, इसी का दूसरा नाम है फिलीफॉर्म एपरेटस. तो ये फिलीफॉर्म एपरेटस आपका रह जाएगा एंड यू विल सी दैट द एग विल ऑल्सो रिमेन अलाइव. ओके, बट जैसे ही आपने देखा कि फिलीफॉर्म एपरेटस जिसका बचा हुआ है, दिस synergids has now been degenerated, so the remains of degenerated synergid will be present there. So you see this is the remains of degenerated synergid, and this remain, it will behave as a chemical signal to the pollen grain. तो हम लोगों ने शुरू में क्या बात किया था कि ये केमिकल डायलॉग है. तो हम लोगों ने देखा कि ऑक्सिन एंड साइटोकाइनिन से पोलन ग्रेन आपके एम्ब्रायो सैक के साथ बात कर रहा था, एंड नाउ द एम्ब्रायो सैक हैज अ डीजेनरेटेड साइटोप्लाज्म फ्रॉम द साइनर्जिड. तो फ्रॉम दिस द एम्ब्रायो सैक विल नाउ गोइंग टू हैव अ कन्वर्सेशन with the pollen grain. So this remains of the degenerated synergid, it works as a chemical signal for embryo sac, right? So isn't it interesting, guys, that the both of these two structures, one is our pollen grain is speaking with the embryo sac with the auxin and cytokinin. At the same time, the embryo sac, once it has degenerated one of the synergids and the remains what is present, through that it is giving chemical signal and speaking with the our pollen grain, which is now has germinated and produced the pollen tube. So what we see that the next we are going to talk about the entry of pollen tube in megasporangia, or what we call as ovule. Okay, So guys, what we have seen so far, that the pollen tube is trying to go towards the egg. So the chemical signal which was coming from the embryo sac is working as a better navigation tool for the pollen tube to finally meet the egg. So we write signals from the embryo sac helps the pollen tube for better navigation. What we see that once it is trying to approach the egg, the pollen tube, इसको सबसे पहले ओव्यूल के थ्रू जाना पड़ेगा और ओव्यूल के थ्रू जाने के बाद फाइनली ये जाके एम्ब्रायो सैक को मीट कर पाएगा. So if we talk about the approach of pollen tube towards the embryo sac, it has to go via the ovule. So we see that pollen tube can enter the ovule from three ways. There are three ways to enter the ovule. तीन जगह है. So we will look at it. So we see that if we talk about an ovule, here I am drawing an orthotropous ovule. So you see that in an ovule there are mostly three directional points. There is one pore-like structure, what we know is known as micropyle. So this is the micropyle region. We see that this is a chalazal or the base region, and there is the presence of integuments which is present in between, right? So we have integument present in between, we have micropyle on top, and then we have chalazal as a base. So what we see very interestingly is that if the pollen tube enters in the megasporangium or ovule from the micropyle end, so what it is, that entry of pollen tube from micropyle end, this method of entry is known as porogamy. So we call it porogamy. If the pollen tube can enter from the chalazal end, we see this is the entry of pollen tube. This is the entry of pollen tube from chalazal or base. So this method is known as chalazogamy, or we call it the basogamy. Okay, and if we see that a pollen tube is entering inside the ovule from the integument, so we see that this is now entering inside the ovule somewhere in between, so we call it the mesogamy, right? So this is the entry of pollen tube to the ovule or megasporangium. So there are three ways to enter inside the ovule or megasporangium. So one is porogamy, mesogamy and chalazogamy. Up next, guys, we are going to discuss about the entry of pollen tube into the embryo sac. Okay. We are going to discuss about entry of pollen tube in the embryo sac. Although we have seen that a pollen tube can enter inside the ovule in 3 different ways, but one has to remember very wisely that the entry of pollen tube in the embryo sac always happens from the site of degenerated synergids. So this is a statement from NCERT. Please remember and write that the entry of pollen tube in embryo sac always occur through the site of degenerated synergid. And the site of degenerated synergid, this is also the site what we call as micropyle. So once we see that the pollen tube will finally reach towards the egg apparatus and it is trying to enter the embryo sac through the site of degenerated synergid, there will be an interaction between the filiform apparatus which is present of the degenerated synergid with the tip of the tube. Okay, so we will see that. So now this is the interaction of pollen tube and embryo sac. What we see here is that, let us imagine this portion is having your synergid. This is the persistent synergid जो degenerate नहीं हुआ था. Then you have the presence of the egg. Then you have the filiform apparatus of the degenerated synergid. और यहाँ पे आपके पास degenerated synergids के जो remains था वो available है. Now what we see is that the pollen tube will come and interact in this region of this filiform apparatus of the degenerated synergids. So this is the pollen tube. तो आपको पता है कि degenerated synergids केमिकल सिग्नल दे रहा था, उसी केमिकल सिग्नल को फॉलो करते हुए हम लोग देखते हैं कि जो पोलन ट्यूब है वो आपके डीजेनरेटेड साइनर्जिड तक पहुंच गया है और डीजेनरेटेड साइनर्जिड का जो फिलीफॉर्म एपरेटस है वो यहां पे प्रेजेंट है और उसी के साथ ये इंटरैक्ट करना शुरू करेगा. सो व्हाट वी सी हियर, ड्यू टू दिस इंटरैक्शन first thing that hydrolase enzymes will secrete from pollen tube and degrade a portion of filiform apparatus ओके. सो पोलन ट्यूब, इसके पास आपको पता है कि इसका जो टर्मिनल रीजन है वहां पे कौन बैठा है, ट्यूब न्यूक्लियस बैठा है. सो दैट ट्यूब न्यूक्लियस जो हाइड्रोलेज एंजाइम सिंथेसिस कर रहा था, उसी हाइड्रोलेज एंजाइम के हेल्प से आपका जो डीजेनरेटेड साइनर्जिड का फिलीफॉर्म apparatus है उसी का थोड़ा सा पोर्शन जो है वो डिग्रेड हो जाएगा, एंड ड्यू टू दिस इंटरेक्शन द एपेक्स ऑफ द पोलन ट्यूब will also degrade. And so we can put it in a different point here, that due to the interaction the apex of the pollen tube will also degrade, and once the apex of the pollen tube will degrade, now you see the pollen tube which was carrying the tube nucleus and the spermatozoids will be released inside the cytoplasm of the degenerated synergid. Okay, so we will draw a last diagram for this topic where we see this was your persistent synergid, जो synergid डिग्रेड नहीं हुआ था, ये उसका न्यूक्लियस है, हमारे पास एग सेल प्रेजेंट है, ये उसका न्यूक्लियस है, और जो हमारे पास साइनर्जिड डिजेनरेट हो गया था इसका फिलीफॉर्म एपरेटस भी है, जो थोड़ा सा पोर्शन हमारा यहाँ पे डिग्रेड हुआ है ड्यू टू दी हाइड्रोलिज एंजाइम सिक्रेटेड बाय पोलन ट्यूब. सो this is our pollen tube which is now have released the tube nucleus. It has also released the spermatozoids in the cytoplasm of this degenerated synergids. लेवल जरूर कीजिएगा. So we see that cytoplasm of degenerated synergid है, ये आपका पोलन ट्यूब है. ओके, ये आपके स्पर्मेटोजोइड्स जो कि आपके नॉन मोटाइल मेल गैमीट्स हैं, and as you see this is your tube nucleus. Okay, so as we see this is the terminal event of pre-fertilization. तो pre-fertilization जो हम अभी तक पढ़ रहे थे, what we see that pollen pistil interaction, this is the terminal event or the last event of pre-fertilization. जो दो आपका मेल गैमीट्स यहाँ पे आप देख सकते हो, हाँ, spermatozoids, they are now released or you can say launched inside the embryo sac, and then just after this incident we will see the fertilization will start. And you have to remember this topic very carefully for NEET examination, and prepare every bit of the details with care. That's all for today guys. Tata, we'll see you soon.